In this video, we are going to have an introduction about biochemistry. Biological chemistry, or simply biochemistry, is the branch of biological science that is concerned with the study of the chemical makeup and functional behavior of living systems at molecular level. Also, it deals with the physiological reactions and biochemical transformations taking place in living organisms and explains the chemical basis of biological phenomena. In recent years, biochemistry has become a most vital tool for the understanding of the physiological and pathological aspects of man and other animals. It is now one of the most exciting and fascinating branches of biological science. The term biochemistry was introduced in the modern sense by Carl Neuberg in 1903. In fact, biochemistry is an offshoot of organic chemistry and physiological chemistry. Broadly, it deals with the complex chemical makeup of living systems and the intricate reactions and complex processes taking place within living organisms. Every life process, however simple or complex it might be, is the ultimate result of a series of biochemical reactions. So, biochemistry is of paramount importance as a pathfinder in unrevealing the chemical mysteries of life and also in tracing or tracking the intricate roots of complex life processes and biological phenomena. Although biochemistry is a relatively new branch of science, most of the questions it attempts to answer are as old as human history. From a modest beginning, the growth of biochemistry has been so amazing and spectacular that it is now a major realm of both biology and chemistry. Recent discoveries and revelations in chemical science have significantly broadened our understanding of the mystery of life. Modern biochemistry consists of two broad areas, namely descriptive biochemistry and dynamic biochemistry. The former deals with the chemical nature of cell components or living systems, whereas the latter encompasses the functional aspects of the molecular mechanisms of energy transactions, chemical regulations, and chemical transformations taking place in living systems. Descriptive and dynamic are the two types of biochemistry. The former, that is the descriptive biochemistry, deals with the chemical nature of cell components or living systems, whereas the latter, that is the dynamic biochemistry, deals with the functional aspects of the molecular mechanisms of energy transactions, chemical regulations, and chemical transformations taking place in living systems. So, first one is the descriptive biochemistry, then the dynamic biochemistry. These are the two types, the major two types of biochemistry. Historical perspectives. Let's learn about the historical perspectives of biochemistry. Until the beginning of the last century, biochemistry was not recognized. Until the beginning of the last century, biochemistry was not recognized as a full-fledged, discrete, and distinctly independent discipline. The first scientific journal published in this area was founded in 1879 in Germany. Three more biochemical journals were published in 1903. They were Germany Biochemical Journal in England and the Journal of Biological Chemistry in the USA. During the same period, the first professorship in biochemistry was established in England, France, and the USA. England, France, and the USA. Biological chemistry, for the most part, is a science of the 20th century. Its probable beginning, much earlier to this, is not quite clear. The reason is that it is basically a synthetic discipline, evolved by the merger of biological science, analytical chemistry, and organic chemistry. Initially, structural biochemistry was indissolubly interlinked with the development of organic chemistry. Studies along the line of modern biochemistry began nearly 250 or more years before. They began more as phytochemistry than as animal chemistry. The first great impetus for the development of biochemistry was probably provided by the pioneering works of the renowned Swedish chemist Carl Scheele in 1742 to 1784 during the 1780s about the chemical composition of plant and animal tissue. He could isolate a number of organic compounds such as citric, lactic, maleic, oxalic, tartaric, and and uric acids, glycerol, several esters, etc. from natural sources. Towards the end of the 18th century, in 1970 to 1972, several early observations on photosynthesis were made by Joseph Priestley, Jan Ingenhousz, and Jan Sonnenberg. With the development of advanced techniques during the early decades of the 19th century for quantitative elemental analysis, the substances isolated by Scheele were found to contain carbon. During the second half of the 18th century and throughout the 19th century, substantial studies have been made to understand the structural and functional aspects of life processes. This was followed by successful efforts to artificially synthesize carbon containing organic compounds from inorganic constituents. It was a rude shock or a death blow to the prevailing theory of vitalism, the concept that organic compounds could be synthesized only through an agency of vital source present only in living organisms. Vitalism was proved baseless by the artificial synthesis of urea from lead cyanate and ammonia by Frederick Wöhler in 18 to 1882 in 1828. The artificial synthesis of urea by Wöhler was a landmark event in the history of biochemistry. The urea was synthesized by from lead cyanate they synthesized urea. The artificial synthesis of urea by Wöhler was a landmark event in the history of biochemistry. It not only disproved vitalism but also gave birth to a new branch of biochemistry which sponsored synthesis of organic compounds from inorganic constituents. Wöhler's work was followed by the synthesis of acetic acid by Adolf Kolb in 1844, the synthesis of several other organic compounds by some other scientists in 1850. By these developments, vitalism was quietly laid to rest. The 19th century witnessed considerable advancements in the structural chemistry of lipids through the outstanding works of Michael Scherl in 1789, 86 to 1889. He revealed that fats are formed from fatty acids and glycerol and also isolated several fatty acids and glycerol. The isolation and characterization of several high molecular weight substances was another significant achievement in the 19th century. Of particular significance were the works of Mulder, Liebig, and some others on the isolation of amino acids, and that of Fischer, who revealed how amino acids are covalently linked end to end to form intact proteins. During the early 1830s, the crucial role of enzymes in biological systems was established beyond doubt. In 1860, the well known French scientist Louis Pasteur did commendable work on fermentation and categorically ascertained the central role of enzymes in all life processes. He also introduced his concept of aerobic and anaerobic organisms. In 1868, Miescher discovered DNA in the nuclei of pus cells and carried out a series of investigations about the properties and distribution of nucleic acids. This was another significant milestone in the early history of biochemistry. It marked the extension of biochemistry to the field of genetics and opened the way to the mysterious world of nucleic acid and molecular biology. The structural chemistry of organic molecules received significant attention in the 19th century. The most significant work in this field was that of the German scientist chemist Emil Fischer, who revolutionized the work on the structure of carbohydrates, amino acids, and lipids. He convincingly established the structure of many carbohydrates, separated several amino acids from the hydrolysates of proteins, recognized the optical configuration of carbohydrates and amino acids, explained the specificity of enzyme action, and postulated the Lock-and-Key hypothesis of enzyme substrate complexing. A breakthrough in enzymology and biochemistry was made in 1907 by Edward Buchner by extracting an enzyme from yeast cells which could ferment sugar. Other significant development of the 19th century were the extensive investigations on photosynthesis and carbon dioxide fixation. The structural chemistry of organic molecules received significant attention in the 19th century. The most significant work in this field was that of the German chemist Emil Fischer, who revolutionized the work on the structure of carbohydrates, amino acids, and lipids. He convincingly established the structure of many carbohydrates, separated several amino acids from the hydrolysides of proteins, recognized the optical configuration of carbohydrates and amino acids, explained the specificity of enzyme action, and postulated the lock and key hypothesis of enzyme substrate complexing. A breakthrough in enzymology and biochemistry was made in 1897 by Edward Buchner by extracting an enzyme from yeast cells which could ferment sugar. Other significant developments of the 19th century were the extensive investigations on photosynthesis and carbon dioxide fixation by Horace Saussure, studies on the process of animal digestion by Lazzaro Spallanzani, Claude Bernard, and others, and the application of surgical techniques for the study of biochemistry and animal physiology. By the end of the 19th century, a new independent discipline was born, and in 1903, Newberg coined the term biochemistry to cherish it. With the dawn of the 19th century, biochemistry burst into full bloom. By 1926, the protein age of enzymes was established, and for the first time, an enzyme urease was purified and crystallized. The invention of ultracentrifuge by Schwarzberg in 1930 opened broad avenues for further achievements in biochemistry. In 1929, Fish and Subaru isolated ATP from muscle extract. In 1932, Lohmann elucidated the scheme of ATP phosphocreatine reaction during muscle contraction, and in 1940, Lippmann described the pivotal role of ATP in energy yielding and energy requiring processes. In 1937, Hans Krebs explained the citric acid cycle of reactions. In 1940, Bedell and Tatum advanced the one gene, one enzyme hypothesis and brought genetics close to biochemistry. This in turn gave birth to the branch biochemical genetics. In 1944, Avery, MacLeod, and McCarthy discovered that DNA is the genetic material and thereby elevated biochemical genetics to a much higher plane. In 1948, Calvin and Benson discovered the Calvin cycle of photosynthesis. In 1950, Kennedy and Leininger revealed that mitochondria are the centers of fatty acid oxidation, cellular respiration, Leininger revealed that mitochondria are the centers of fatty acid oxidation, cellular respiration, and ATP synthesis. In 1954, Daniel Aaron and his team reported the synthesis of ATP in chloroplasts by photosynthetic photophosphorylation. In 1953, Watson and Crick explained the molecular organization of DNA, opening a new era of exponential growth of biochemistry, molecular biology, and genetics. In 1956, Arthur Kornberg discovered the enzyme DNA polymerase. In 1957, Sutherland discovered cyclic AMP, elucidating the regulatory mechanisms in biochemistry. In 1958, Ingram revealed that normal hemoglobin and sickle cell hemoglobin differ from each other with respect to only a single amino acid residue. From 1968 onwards, biochemistry began to acquire an interdisciplinary status, sharing much in common with molecular biology, molecular genetics, and molecular biophysics. The year 1961 witnessed several landmark developments such as the polyuridylic acid coding for phenylalanine by Nirenberg and Mathai, discovered allosteric enzymes by Jacob Monod and Cengiz, and the elucidation of the genetic control of protein synthesis by Jacob and Monod. By 1965, genetic code was well established, and the role of individual codons in protein synthesis was explained by Nirenberg, Khorana, and Holle. Towards the close of the 20th century, significant achievements were made in the fields of immunochemistry and biochemical engineering. In 1975, Milstein developed hybridoma cells and produced monoclonal antibodies which could be used for the purification of interferons. In 1980, Snell, Benesharaf were awarded the Nobel Prize for their outstanding contribution to the biochemistry of immune systems, and Paul Berg was awarded the Nobel Prize for his contribution to recombinant DNA research. Paul Berg was the first to splice together genes of different species. The period 1981 to 82 witnessed the first ever synthesis of the alpha interferon gene by a group of scientists in Leicester. The growth of biochemistry during the past 3 decades has been exponential, and its achievements amazingly spectacular. Biochemistry has now crossed all conventional boundaries between the different disciplines of science and has become an important, all embracing multidisciplinary branch of science with unlimited applications. Scope of biochemistry. Up to the 1930s, the central theme of biochemistry was immediately intermediary metabolism, that is, the elucidation of the intracellular synthesis, synthetic and degradative pathways. But at present, it has significant roles in several major areas. Some of such areas are the following: intermediary metabolism, physical chemistry of biomolecules, mainly proteins, enzymes, and nucleic acids, organic chemistry of enzyme catalyzed reactions, biosynthesis of proteins and nucleic acids, bioenergetics, especially oxidative phosphorylation and ATP synthesis, cellular control mechanisms, ultrastructure and molecular architecture of cell, molecular basis of genetic and developmental process, molecular basis of physiological phenomena such as transmission of nerve impulses, muscle contraction, vision, transmembrane transport, etc. Biotechnology, especially recombinant DNA technology. In term Up to the 1930s, the central theme of biochemistry was intermediary metabolism, that is, the elucidation of the intracellular synthetic and degradative pathways. But at present, it has significant roles in several major areas. Some of such areas are the following: intermediary metabolism, physical chemistry of biomolecules, mainly proteins, enzymes, and nucleic acids, organic chemistry of enzyme catalyzed reactions, biosynthesis of proteins and nucleic acids, bioenergetics, especially oxidative phosphorylation and ATP synthesis, cellular control mechanisms, ultrastructure and molecular architecture of cell, molecular basis of genetic and developmental process, molecular basis of physiological phenomena such as transmission of nerve impulses, muscle contraction, vision, transmembrane transport, etc. Biotechnology, especially recombinant DNA technology. So this is all about the introduction of biochemistry.